Hello everyone, welcome to a new session on molecular biology. Here we discuss side-directed mutagenesis. The generation and characterization of mutants is an essential component of any study on structure-function relationships. Knowledge of the three-dimensional structure of a protein, RNA or DNA can provide clues to the way in which they function, but proof for the fact that the correct mechanism has been elucidated requires the analysis of mutants that have amino acid or nucleotide changes at key residues. Mutants are generated by treating the test organism with chemical or physical agents that modify DNA. This method of mutagenesis has been extremely successful as witnessed by the growth of molecular biology and functional genomics, but suffers from a number of disadvantages. First, any gene in the organism can be mutated and the frequency with which mutants can occur in the gene of interest can be very low. This means that selection strategies have to be developed. Second, even when mutants with the desired phenotype are isolated, there is no guarantee that the mutation has occurred in the gene of interest. Third, prior to the development of gene cloning and sequencing techniques, there was no way of knowing where in the gene the mutation had occurred and whether it arose by a single base change, an insertion of DNA or a deletion. As techniques in molecular biology have developed so that the isolation and study of a single gene is possible, so mutagenesis has also been refined. Instead of crudely mutagenizing many cells or organisms and then analyzing many thousands or millions of offspring to isolate a desired mutant, it is now possible to change specifically any given base in a cloned DNA sequence. This technique is known as side-directed mutagenesis. It has become a basic tool of gene manipulation, for it simplifies DNA manipulations that in the past required a great deal of ingenuity and hard work. For example, the creation or elimination of cleavage sites for restriction endonucleases. The importance of side-directed mutagenesis goes beyond gene structure-function relationships, for the technique enables mutant proteins with novel properties of value to be created by protein engineering. Such mutant proteins may have only minor changes, but it is not uncommon for entire domains to be deleted or new domains added. SDM is used for investigating the structure and biological activity of DNA, RNA and protein molecules and for protein engineering.